Now in this video, we are going to detect the analysis of contrast in visual images. What happens actually is, for example, this is a blank wall and you are staring at it, then only a few neurons will be excited. It means that whether the light of this is, well, like if it's light or if it is dark, but in both the cases, only a few neurons will be excited. But if there is an image, like the image, like this, what will happen in such a situation that along the borders of this image, there will be maximum stimulation. You can see the maximum stimulation. So why is that so? At the border, what is happening? There is change of color from one color to another color, from light to dark, from light to dark, or from dark to light. There is a change, there is a transition. So the intensity of stimulation of the neurons, it depends on the gradient of contrast or the amount of contrast or the amount of change. So change causes stimulation. Greater will be the sharpness of the contrast, greater will be the color difference, greater will be the intensity of stimulation. The greater is sharpness, greater is intensity difference, greater is the stimulation. So in a visual image, the visual cortex not only analyzes the direction of a line, whether it's vertical, whether it's horizontal, whether it's diagonal, but this thing is all recognized. Not only the existence of these lines and border, but the visual cortex also analyzes whether it's vertical, horizontal, whether it's diagonal. It is because our neurons, they are arranged in vertical columns, and whenever there is this different stimulation will cause different region of the vertical columns to be activated, and that is how this differential activation leads to recognizing the patterns. Also, they are telling you whenever there is a contrast edge, the second order neurons, the inhibiting neurons, they are going to cause inhibition of the cells. And when there is inhibition of the cells, what will happen? We will recognize that for example, the whole retina is activated, but this thing, this pattern, all of those are inhibiting. Those inhibiting regions are further going to cause signals to be stimulated, and these inhibiting signals, they're majorly the signals which are going to detect the line pattern. They are called simple cells, and they majorly activate the region number 4 of the primary visual cortex. Now the simple cells, they were recognizing the line, the orientation of the line. Now we have complex cells as well. These complex cells detect the line orientation. Also, they also detect, so for example, there are a large number of the lines. So they are telling you that, as I told you, there were like 6 regions of the visual cortex, right? And in the layer number 4, you have further 4 differentiation. Now they're telling you that from when you're drifting away outwards, like inwards, like 1 to 3 layers, which are outwards, or like 5 or 6 layers, which are like inwards. Whenever you're moving towards these layers, what will happen? That these layers will help you to recognize that this large number of the lines, whether they are oriented in the same direction or not. If they are like this or something like this, it means that they're not oriented in the same direction. But if they are oriented in the same direction, this will be done by the layers that is closer to region number 4, and they will help us to recognize whether these lines are oriented in the same direction as the first line or not, and that is depending upon the complex cells. That helps us to recognize various different lines and their correspondence with each other. Now they're telling you that in the outer layer of the visual cortex or in the secondary visual cortex, what are there? There are specific areas which are only stimulated by certain lines of certain specific lengths, by specific angles, or by specific shapes. These neurons are going to detect higher order of information. They're going to help us to analyze the further discrepancies of the image. They're going to further analyze the characteristics, like if this is a circle. Okay. So firstly, the simple cells are going to recognize whether there are any borders in it, whether there are any lines in it. So simple cell thing is done. The complex cells, they are going to recognize if they have any lines or borders, whether they are oriented in the same direction or not. And then further going ahead in the most outer region of the visual cortex, they're going to recognize whether they have certain borders, whether they have certain angles, whether they have a specific shape. So the shape pattern that is done by the outermost region of these neuronal layers or by the secondary visual cortex. And then further moving into the layers, they are going to help us to decipher what is the shape, what is the meaning, what is the visual information that this particular circle is going to give us. Now, detection of the color. It is done in the same way as I told you that the color is detected by color contrast. Whenever there is a change of color, whenever there is a gradient of a change of color, that is going to cause a stimulation. And how is this difference taking place? It is green area is alternating with the blue one, red area with the green one. Majorly it is green or blue area with the red one. So G or B is contrasting with the red one, or green is contrasting with the yellow one. All these colors can be differentiated against a white area as one, but majorly they are, this is how the differentiation takes place. But all the colors, like all the colors of the prism, they can be differentiated against a white light because that helps in the maximum contrast being built. This mechanism depends on opponent colors. This is the main mechanism, and I told you that these colors were like opponent colors, so that is how they are going to activate different regions of the visual cortex. And more complex is the contrast, more complex will be like the degree of stimulation. Like a simple contrast, it is detected by simple cells. Complex contrast, it is detected by complex cells or hypercomplex cells. If like you have green alternating with red, it will be detected by simple cells. But if you have a green alternating with a purple, or green alternating with a cyan color, or green alternating with a also you can say mauve color, all of those colors will be detected by complex or hypercomplex cell, because that type of thing is associated with some different cell, that is a complex contrast. Now what will happen if you remove the primary visual cortex? You will have loss of conscious vision, that is blindness. You will not be able to consciously look at certain object. But if the superior colliculus is intact and the visual primary visual cortex is gone, then you will be able to react subconsciously to change the light insights. For example, you have your eyes and there is some, in the lateral portion of your eyes there is some visual disturbance, but your primary visual cortex is not intact but the superior colliculus is intact. What will happen? That you will be able to respond to that light. If that light is sharp, you will close your eyes, you will move your eye or body towards or away from that object. So turning the eyes, turning the head, turning the body, all of that is controlled by the superior colliculus. Now we have field of vision or perimetry. Field of vision, as you know, your eyes, they have a medial side and a lateral side. Medial side because it is toward the nose, it is known as nasal side, and lateral side because it is toward the temple region, so it is known as the temporal field of vision. So to recognize the blindness, what you do is like you have your eyes over here, you call the person to recognize or fix the eyes on that particular object, and after that you're going to have a certain visual disturbance like a light, and you will move it from here to here, and then you will find out the blind spot in that eye. If the blind spot is at the level of the optic disc, which is the physiological blind spot, it means that you are fine, you are perfectly fine. This optic disc or the physiological blind spot in the eye is the area which lacks any rods and cones, and it is present like 15 millimeters from the fovea. But when you have abnormality of vision, you do perimetry, you call the person to fix your eyes, and then you do the blind spot test, and the blind spot test comes out to be from another region rather from the optic disc, that it means that there is some abnormality. It is known as scotomata, that is known as pathological blind spot. It is majorly the scotomata or the pathological blind spot. It is caused due to damage to optic nerve, majorly caused by glaucoma, which is increased intraocular pressure due to the disturbance and drainage of the aqueous humor. Then there can be allergic reactions, then there can be toxic condition like lead poisoning, then there can be like cigarette poisoning, etc., excessive use of tobacco. Now we have retinitis pigmentosa. What is that? It is a degeneration of various portion of retina. It is caused by excessive deposition of melanin. Like in the retinitis pigmentosa, you will see certain black colored dots or different colored dots deposited or made in your eye because of the presence of melanin pigments. And these pigments are made in the scar area or degenerated regions. What will be effect of these? First of all, since they are present in your eye, they are going to cause blindness in the peripheral field of vision, because these spots, they are going to uh, damage the transparent cornea. So firstly they are going to damage the peripheral region, then they go towards the central region. So retinitis pigmentosa is generally degeneration of different portions of retina that spreads from the peripheral to the central portion, and the degenerated portion of the retina, they are replaced by melanin pigment. That is all from my side regarding this. In the next video, then we are going to talk about the eye movement and their control.